And now, to be wowed again, here's Erinn Muller.
Good afternoon, all. I'm Dr. Erinn Muller. I'm from Mote Marine Laboratory, so I'm a local gal here. I've been here a little over a decade and really excited to share what we're all about with coral reef restoration and innovation in Florida's coral reef. So a big shout out to you all. You guys are the diehards. We've made it this far. This is the last talk of the day, and I will make it as exciting as possible. I promise. Okay, once the slides come up. So I'll get us started. Mote Marine Laboratory is a completely independent nonprofit research institution, and it's actually just right across the bay over there. That's our original mothership, as I call it. Look. Here we go. And so I'm sure many of you, if you're local, you have visited this place. It's obviously not poised to deal with climate change well, including storms and sea level rise, which we experienced multiple times last year. And so although this is going to be a new innovation center for research down the road, and we still inhabit this location and do a lot of cool research here, our new mothership, literally, is right also down the road at UTC in Bedresson Park. So a beautiful new public science education aquarium. We have lots of corals in that location as well, and I encourage you all to visit it. Although those are our two main public locations, a lot of our coral work happens outside of those walls. So we actually have an incredible hub of coral work happening out on Fruitville Road at our Mote Aquaculture Research Park, which I will talk about near the end of my slides. But the main hub of activity is Mote's Elizabeth Moore International Center for Coral Reef Research and Restoration, so aptly named after the woman we all know and love, who's in the room with us here. It's in Summerlin Key, Florida. It's this insane infrastructure, 19,000 square feet of research, classrooms, dorms for visiting scientists, and it's right on Florida's coral reef. So we can access it literally out our back door. It's also one of the largest land-based nurseries that surrounds this infrastructure as well, which I'll show you some up-close and personal images of. Okay, so why are we at Mote focusing so much on coral reef ecosystems when there's so many different marine ecosystems that are incredibly interesting and valuable? And it's because of the dire loss that we learned about earlier with Dr. Harborn's presentation, that we have lost over 98% of our living coral in Florida's coral reef. So although this used to be an iconic location for people to come visit and dive in our coral reefs, this is now, unfortunately, an iconic location of reef lost. And our story of loss is kind of a poster child of what the rest of the world needs to prevent from happening. And although there is a large and vast network of marine protected areas throughout Florida's coral reef, which spans from Martin County on the east coast all the way down to the Dry Tortugas, and all those pretty little lines represent different managed areas, which are protected in one way, shape, or form by different management agencies. Although those have been in place and are well managed, we have lost these corals, and there are no signs of natural recovery because of the issues that are happening are well outside of those management area capabilities, unfortunately. Climate change, major disease outbreaks, and a lot of things they just can't control. But because of that, people at Mote and at lots of different organizations around Florida over 15 years ago recognized that these corals were not coming back on their own. And over the last decade or two, we as a community have created an extremely effective network of coral restoration nurseries throughout the state. So these are run by over a dozen different institutions that are universities, nonprofits, state agencies, who else do we have up there? Some education groups. And they have contributed to create the reality of bringing that reef back through the process of restoration. And so now we have over 14 different in-water coral nurseries, and they look something like this. We have these awesome underwater coral trees where instead of branches of pine needles kind of hanging off, you have corals dripping off of them, like growing beautifully underwater. And we also have a lot of these land-based nurseries throughout Florida as well. And so these are tanks on land, so they're more accessible to the public. You can control the environment. If kind of crap hits the fan out on the reef, you still have things on land in holding, for safekeeping. And in addition to these nurseries, which are focused on propagation for restoration, we actually have built 2 solely dedicated infrastructures based on coral biobanking. So making sure that we hold these corals indefinitely to safeguard those species. And I'll get a little bit more into that later. Okay, so everybody does restoration a little bit differently, but we all kind of follow the same sort of cycle. So I'm going to kind of walk you through what coral restoration at Mote looks like in the face of climate change, right? But to kind of orient you into this process, we'll take it step by step. So the first step for coral restoration really started with asexual fragmentation. So asexual fragmentation is like a cutting. If you want to kind of get that another propagule of that plant, you just snip that little piece off that plant. Now you have 2 of them, right? And you can grow more individuals. So corals can do the same thing, even though they're animals. Remember, Alistair talked about how they're made up of these little polyps that all work together. Each of those little circles kind of on those ceramic discs represent a coral colony, and the little circles in there are those polyps. As long as you have a cutting of 1 polyp, you can make more corals indefinitely through time through that asexual fragmentation process. So this is kind of a view of in one of our raceways, and you're looking at lots of different kind of propagules of coral fragments that are ready to be outplanted onto the reef. So how we make more of them, though, is just by this process that we call microfragmentation. So we take a big piece of coral, or we can take a little piece of coral. As long as it has one of those polyps on it, we can make lots of little ones that are like 1 centimeter by 1 centimeter square, put them onto new ceramic discs, and use that propagation pipeline to make a lot of more biomass quickly. And what we actually see is these corals grow really fast when you use this process of kind of cutting, because it triggers a growth response. So we can get a lot of corals from a little bit of biomass within a short period of time using this process, which has been really useful to apply for things like boulder corals and brain corals, which grow really slowly. Now, the little image over here on the right shows what we do for the branching corals, the staghorn and the elkhorn coral. So these grow really fast and they love those trees. So it's a little bit easier. You can kind of snip a little branch off, hang it up on the tree next door, and we keep kind of propagating those corals over and over again, so that we always have a lot of biomass to get back out onto the reef. Okay, so once we make those corals, now we've got to grow them out a little bit. So we don't want to cut them and put them right back out because they're very stressed out. So we grow them within our underwater nurseries. And I mentioned kind of what these look like before. Here's an overhead image of some of our raceways outside of our Elizabeth Moore International Center for Coral Reef Research and Restoration, which we call IC2R3 for short. If I say that again, I will use that one. And so you can kind of see what it looks like. There's these racks and racks of corals that we grow until they are about 3 centimeters in diameter, and we'll put them out onto the reef at that point. And we outplant about 20,000 a year from this process alone. And then we have multiple of these in-water nurseries that we have at Mote. We can grow these beautiful branching corals. We can grow the boulder and brain corals that we have over here. And we can outplant about 30,000 corals a year from these underwater infrastructures. Okay, so how do we actually outplant them? That's a question I always get. And so we outplant them using monogenic clusters. So we talked a lot about DNA and Liv's talk. What that means, monogenic, is every single fragment came from the same individual to begin with. So all of us in this room here, unless there's 2 identical twins out here, which I don't see right now, we all have different DNA, right? And so if you and I tried to merge our tissues together, they would reject each other. But an identical twin would recognize you're the same as me, like let's merge. And that's what corals do. So we outplant them in clusters, and then they start growing over time. And as long as they were the same DNA, they'll touch each other and say, oh, you're just like me. Let's become one. And they do. They fuse together and they create one big coral over time. And so this was an overhead image of like what it would look like on a ceramic plate, but this is exactly what happens out on the reef. We outplant these discs of corals made through the microfragmentation process. They eventually grow out, touch each other, and then fuse. And then this is like a little 1-year-old elkhorn coral that we created through this process. It's probably about 1 year after outplanting. So that's kind of the methodologies we use to grow these corals to get a lot of biomass kind of out on the reef as quickly as possible. But, those corals out there are, subject to all of the issues that got us here in the first place. So putting a bunch of corals out there and just hoping for the best is not the best option, right? So that's where a lot of my research comes in. My work focuses on understanding how to make these populations more resilient. And of course, today I'm going to be focusing on climate change. So 2023 was a real eye-opener for us down in the Florida Keys. It was the warmest year on record. This is kind of a confusing figure to look at, especially this late in the day. Basically, the purple line is the actual sea surface temperature, and these colored areas is heat accumulation in the ocean. Now, corals can withstand warm waters, but only for short periods of time. And so if it's warm for a long period of time, they're accumulating heat stress, which kind of makes these colors darker. So this is kind of a typical year for us. This was 2022. Now, this is what 2023 looked like. So it was got hotter earlier. It was more intense. It lasted longer than anything we've ever had to deal with before. And we knew we were working towards facing a future that looked like this, but I will tell you, this one took me off guard. It happened a lot sooner than I thought would happen. But it was a great natural experiment that we are learning a ton from. But when you go into the ocean at this time, this is what the reefs in our nurseries looked like. So our outplanted corals or the natural corals were all completely bleached. The nurseries obviously could not escape that were in the water. We did try to do a mass evacuation, which did save some, but was extremely challenging. But basically this led to the functional extinction of all of the staghorn and elkhorn corals throughout the entirety of Florida's coral reef. So at this point, we only have about 37 elkhorn coral left on the reef and probably about a similar amount, maybe a little bit more of the staghorn. But I do have we do have signs of hope that I'm going to be sharing with you. But this was extremely intense event, right? Which makes the lessons learned the most important. So as I said, we lost a lot of those corals that were out there. And this is an example of the mortality rates of corals that were outplanted that were strictly the staghorn corals. So these are the branching corals. They grow really fast, but they're also very susceptible to high temperatures. And the high amount of red you just see is like high mortality at those outplanting sites of that species. But our boulder and brain corals actually did remarkably well, where we had over 75% survival. So these corals, were very inherently heat tolerant. Even the biggest event that we'd ever seen and never predicted would happen so quickly, they did relatively well. However, you can see the different kind of shape that these corals would provide to the reef. The one on the right is going to provide lots of different habitats for all of the fish, that Alistair needs to do his work, right? So they need all of that habitat niche space in order to hide from predators or ambush predators or to create that biodiversity, where you're not going to get that same level of kind of interaction with some of these boulder corals. So that's why it is so important for us to understand how we increase the heat tolerance of those corals that we're putting back out onto the reef. So what we do is we take all of the corals that we are propagating for restoration and screen them to tolerance to different threats. So obviously heat tolerance is our big focus for today's talk. And you can see an example of these are 3 corals of different genotypes that were under the same temperature conditions, where the one on the right looks really healthy and the one on the left looks completely bleached and is going to die. So we want to use more of the one on the right, when we're thinking about placing corals out that are going to be able to withstand these high water temperatures. And so we are constantly going through our corals to screen them and understand what makes each one special, which ones are going to be heat tolerant, which ones are going to be resistant to ocean acidification, disease resistant, and to understand within our population of restoration, how can we use that information to do better as we move forward. One of the challenging things is these experiments take a ton of time, and you invariably end up killing the corals in the process of learning. And so it hinders on your biomass and your production. So one of the projects that we're doing is a collaboration with Dr. Kenneth Hoadley, and he is developing a tool and using AI and machine learning to predict heat tolerance without ever having to do an experiment, which is phenomenal. So this is some of the data that we're using to build the algorithm and to make the predictive models. These are 60 different genotypes of corals, and this is their response to heat stress. So the ones that are more negative bleached more, so they're more susceptible. And so they look something like this. The one on the right, those corals are bright white. The one in the middle, they're kind of pale, and the ones on the left are like nice and dark, right? So we are using this information to train his machine that basically you can like do a little fiber optic, little zappy zap, and it tells you all of this data, and it's going to say, this coral is going to be more likely to be heat tolerant. This one's going to be more likely to die in the next bleaching event. And we can do that on thousands of corals basically a day so that we are not bottlenecked by some of our research, which is pretty awesome innovation that we cannot wait to get online. Now, I talked a lot so far about genotypes, but remember, corals have algae that live inside their tissue called zooxanthellae, and those are incredible sources for heat tolerance. So some of them actually are more heat tolerant than others, and corals can be flexible with what algal symbiont they may have. So we kind of noticed through some of our research with Dr. Courtney Klepek, who's at Stanford, that some of our corals that have the algal symbiont, so this is like a cool picture of a polyp, it's in its little coral light structure, and all those little dustings are the algal symbionts, right? And that's what feeds the coral. And so when those corals had the heat-susceptible algal symbionts, they died, basically, on that top row. Now, some of the corals could shuffle. Like I said, they could shuffle to a different algal symbiont and bleach and recover, and they acquire the heat-tolerant symbiont, and then some actually already had that. So right now, we are doing a series of experiments to figure out how do we encourage the acquisition of these heat-tolerant algal symbionts and then get them to be maintained out on the reef. And so that is a huge area of research that we're leading with a lot of different groups involved to help us push that needle forward to make sure we can increase resilience to heat tolerance and climate adapt these corals. And this is kind of just a quantification of that increase in heat tolerance with these heat-tolerant algal symbionts. So this was a project we did with Dr. Rich Karp, who's at NOAA, and he basically compared how much more tolerant these corals are if they have the heat-tolerant algal symbiont. So basically the green dome on the top is the bleaching susceptibility level when you have the heat-susceptible algal symbionts, and then the one on the right is when you have the heat-tolerant algal symbionts. The big take-home message is basically it gives you a 2 degrees Celsius increase in heat tolerance, which could make or break these corals' survival over the next decade, right? So this is incredibly important information that we can use to do tangible action today. And it's not just the genes of the corals or the algal symbionts they have, it's actually what kind of coral it is. So a lot of the corals that we are using now for restoration they're old dudes. They've been around a long time. Who knows how old they are, because corals they hide their age like every woman wish they could. And so we are working with sometimes corals that could be thousands to hundreds of years old. We really don't know. And it looks like when you outplant those corals, they may have a higher susceptibility to things like climate change. So again, these are outplants of some of our branching corals, our staghorn and our elkhorn corals. And this is just like survival prior to that 2023 bleaching event. So it looks pretty good. Everybody's on the up and up. We're getting really good survival. But then the 2023 bleaching event hit and we had really high mortality, right? And these graphs this is on the scale of 0 to 2%, and this is 0 to 14. So very, very high survival. But the thing is, the ones that survived were babies that we created through sexual reproduction. So the next generation of corals that we're putting out there are surviving a much better than their parents. There's lots of different reasons this could be happening. Again, this is another area of research that we are exploring to figure out why. Is it because it's a fresh new genetic combination? Is it because they're able to kind of adapt into this new world? Lots of areas for open-ended questions that we are digging into. So let's talk about our sexual reproduction efforts. So we did talk about asexual, but we like to make babies too. And coral sex is crazy. So we grow corals to be a certain size. They reproduce based off of size, not age, and they need to be about a dinner plate size in order to make babies. So we have areas where we dedicate these trees to make big corals for spawning purposes. We take them into the lab in about July of every summer, and then we hover over these tanks like crazy people throughout the entire night until spawning happens. And spawning looks something like this, where you have these are actually hermaphroditic corals. So these are kind of egg sperm bundles that are released. It's like reverse snow happening, right? And then we pick each coral each gamete bundle up by hand, basically, as it's coming out. We keep track of who came from where, because we do a lot of managed breeding. But this is wild because corals will spawn one night out of the year, basically. And it's for this species, it's about the third to fourth night after the full moon in August, a couple hours after sunset, right? And so, and like Liv was saying, like if that just doesn't happen, if something's off, then you are messed up for another year, right? So this capturing kind of this event is always magical and stressful, but very impactful at the same time. So what do? We take those eggs and sperm and we mix them together and we do assisted sexual reproduction. We settle those babies in the lab and we grow them out for restoration purposes. But what we do is a managed breeding program. So because we know these parents, we know who's heat tolerant, we know who's disease resistant, we know who's highly related to somebody else. We can take parent 1, cross it with parent 2, and then make a bunch of babies that, if these traits are indeed heritable, should be passed down to their offspring to help us increase the resilience of that restoration population. And if you remember, like Biology 101 and Punnett squares, we live this world, which is terrifying because I never thought I would be kind of revisiting this stuff after the P's of Mendel. But here we are. And so literally every single spawning season, we have our list of different genotypes, what we know about them, and we make these like squares about what kind of babies we could create if these corals spawn. And these are kind of our target priorities for every single spawning season. And just to be, because spawning's cool, this is like what a restored boulder coral spawning event looks like. So this was in 2020, where we had 80% of our outplanted corals spawn in synchrony with the wild population, and our outplanted corals survived lots of extreme events like mass coral bleaching, hurricanes, stony coral tissue loss disease, and more bleaching. And so, this gives me hope, right? There is foundation within doing the science behind the effort, and we can create some of these really impactful contributing corals to bring them back to the reef to help us jumpstart that population recovery. But we have a lot of reef to recover. And so there's 365 miles of reef to take care of. And so what we are doing is trying to integrate automation into the process, right? Right now, everything we touch, every single coral, every day, we outplant it every single day by hand. And we really need to work on automation so that we can do more faster. And so we are working with a series of teams that are doing things like automating the monitoring process, the coral health screening process, and the propagation process. Maybe even being able to use robots to outplant the corals for us if we can, kind of figure out how to make that happen. We do some cool stuff with structure from motion technology, where this is like coral reefs in space. So on the left, you have a pre-outplanted coral reef, and on the right, you will see these staghorn corals popping up throughout the reef. This is like one afternoon of effort a year before this kind of took place. And so you can see all of these novel corals popping up and how impactful that can be. And so we're using this technology to monitor things like coral cover, recovery, how much is changing the rugosity, so like how much structure it's creating through time, how these corals are growing, and so forth in three-dimensional space, which is pretty cool. And just kind of give you an overhead image of what this looks like and how we are trying to integrate AI. So we have every outplanted reef basically imaged like this using 3D photogrammetry, and we can put that into 2D space. And then we are hoping to use AI to automatically track these corals through time. So right now we literally have to hand, snap in this information. And really this is a tool that AI could be using to help our staff do better things with their lives than tracing corals on a screen. And so I think, there's a lot of training that's happening now, but I think that's going to be a really effective tool for us to integrate into figuring out how to do restoration even better. And now I've got two side hustles I want to share with you that I think are really important. In addition to this entire cycle, which we are doing on a day-to-day basis, we've started integrating herbivory restoration into our restoration process. This is led by Dr. Jason Spadaro, and it focuses on this crab right here, the Caribbean king crab. And so, reefs, as we learned earlier, are heavily dominated by a lot of this fleshy algae, which is bad for corals because we've lost things like urchins, right? So we've had a lot of urchins lost, which you did a lot of that reef cleanup. But in addition to urchins, these crabs actually eat a ton of algae, and we haven't really been utilizing them in some of this herbivory recovery. And so now we are kind of taking a very dirty reef, basically with a lot of algae, adding crabs, and it can clean up the algae for us while we're doing the restoration process. So they're the maintenance crew that we're putting out there, to make sure that the reef is healthier than before. And we have a huge crab hatchery out at our aquaculture research park where we are growing these baby crabs that we are going to be putting out onto the reef as part of our normal restoration operations to help increase in that cleanup crew. Mm-hmm. So basically we'll take corals, we'll put them out there, we'll put out more, and we'll add crabs, and we'll save the reef. Okay, so in addition to this, I just want to highlight our living coral biobank. It is at Mote's Aquaculture Research Park. It is an amazing facility that's really focused on saving coral species from extinction. So right now we have about 28 different species, representatives from the Caribbean and the U.S. We track every individual coral. We do living coral biobanking. So these corals are there, they're alive, they're growing. We also, I'll come back to that. We also do cryopreservation, which Liv mentioned. And so we are cryopreserving sperm, not just for our own propagation uses. So we can make coral babies with that cryopreserved sperm, but we also deposit it into the USDA germplasm facility for that long-term conservation tool. And I will say, I mentioned before that we lost all of the staghorn and elkhorn corals throughout all of Florida's coral reef in 2023, minus about 37. But because we had the gene banks in place, both us and the Reef Institute, which is in West Palm Beach, another great organization, all of the corals' genetic diversity was saved. Every single genotype that was there before 2023 still exists. Thank you. It is definitely I think it's the most important thing we are doing is just preventing the loss of these species, and super stressful, but very important. Okay. And we do it with a global network. So we're actually part of the Global Coral Biobank Alliance. We work with people in Australia, in Saudi Arabia, Monaco, the Great Barrier Reef, the AZA facilities throughout all of the U.S. And so it's a huge global network working together, not just to save corals in Florida or in the Caribbean, but globally. So we're hoping within the next several years, we'll have every single coral species banked within a global biorepository for safekeeping. And we talked about collaboration. I just want to say it takes a village to do this stuff. We at Mote have great scientists, but we couldn't do it without our collaborators. Here's just some of the people we're currently working with on some of our projects focused on restoration, climate change resilience, and adaptation. And I just want to have a big shout out to probably my favorite project going on. It is called the Reef 5 Hub. It is 9 different institutions in Florida working together to integrate climate resilience and adaptation into coral restoration. So it's a first ever kind of collaborative effort for anybody who's doing this kind of work to do it collaboratively, to do it together, and to be cohesive so that we can be greater together, than the sum of our parts individually. So it's an amazing collaboration and one that I'm very grateful to be a part of. Okay, so we're all wrapping it up here, but I wanted to end it with just saying, there are ways for you to get involved and to make a difference on a day-to-day basis. So this is something I always try to share whenever I do a public talk. This is the International Coral Reef Society 12 pledges for anybody can, do these to help coral reefs. It's, supporting policy, reducing your carbon footprint, and promoting coral reef education and conservation. So this is a great kind of, how do I do this on a day-to-day basis? Here are 12 things you can do. If you want to get involved at Mote, we take people out with us, diving in the keys to do restoration with us side by side. So if you are a diver and you want to go check that out, here are 2 different options for you to explore to get hands-on opportunities underwater doing coral restoration. We obviously have a lot of volunteers here in Sarasota, also down in the Keys, directly within our coral programs. There's lots of opportunities. If you don't mind getting your hands wet, we have lots of things for you to do. You can also tour any of our facilities. So we have tours out at the Aquaculture Park and all of our facilities down in the Keys. You can get hands-on experience on land where we teach you how to fragment corals. It's called Slice of Paradise. So if you want to do that, you can always sign up. We teach you how to microfragment. You can touch the corals. You create new corals through that process, and it's really, really fun. And if you are a Florida registered car driver, you can get a Protect Our Reefs license plate. So this directly supports Mote's coral reef research and restoration activities. I have the coral plate. It says coral on it, so you can't get that one, but you could get something else within your vanity plate that would be just as cool. And then finally, of course, you can donate. So if anybody is interested in donating at Mote or one of our coral restoration organizations, here's the QR code to directly do that. And with that, I will take questions.
We'll have this one up front, and then I'll grab you in the back.
Thank you. I feel overwhelmed, but at the same time, I've been a person involved in education for a long time. Can you talk a bit about the career path, the workforce that is with you now, and what are the opportunities? And hopefully it's a transdisciplinary, that's the word I use in healthcare, approach to trying to build the next workforce to address all that you've shared.
Yeah, so we have a lot of great opportunities at Mote for workforce development training, specifically with coral. So we have an internship program that happens year-round, and we have interns that can be kind of conventional or unconventional in that process, and they are undergraduates or graduate students. We also have high school internship programs, too, that people can sign up for. So I actually have one high school student right now. She's working in our coral workforce development lab out at our new aquarium, and she's in there multiple times a week doing some research projects, learning how to take care of corals. So it's hard to find somebody that knows how to do this, right? So this is a direct pathway to get a job at one of those 14 different institutions around Florida that are directly involved in this type of work. And I will say a majority of the people that we end up hiring at Mote are former interns. It is a great pathway to get your foot in the door. And I think there's a lot of potential, for people to learn and do that. And I would say a lot of these groups that we work with also have a similar internship program.
I will being a that there must be a work to work.
Yes. Yeah. So, right now we have some staff that really focus on a lot of data analysis and data interpretation. I also have this amazing volunteer who lives in Colorado and is a data scientist, and he's got time on his hands. So I send him data all the time, and he sends me back these R scripts, these novel apps, and all the information associated with interpreting the data. And people like that are crazy amazing because they are so smart, and they are volunteering their time, and they're helping us make headway on things we don't have time to deal with. Perfect. I love that.
Okay, Erinn, right back here.
Hi, this might be a silly question, but as someone with a home on the water, I have one of those little artificial reefs under my dock. I have oyster strings. Can I do coral trees?
Well, there are corals that live locally, which are wild. I was just out a couple days ago snorkeling with DEP, looking at corals for us to collect for gene banking purposes here. And you would have to have specific permits that I would not be able to share exactly what they would be. But there are corals that live here locally in Big Pass, New Pass. If you snorkel around, you'll see stony corals. There's specific kinds that only live in these turbid waters, with high fluctuations of temperatures, but they are very hardy, and they could live right on your dock too.
Any other questions? Time for one more question if you have a question. If not, we'll get ready for Bob and our discussion.
So the one thing I want to do while we put some chairs up on the stage for our last panel, and Erinn, that was amazing.